This is going to be cell respiration part 6, other metabolisms. Throughout this whole lesson of cell respiration, we have been breaking down sugar. You guys don't just eat sugar. Obviously, it is a big component of your diet. Fruits, vegetables, bread, grain, sweets like candy, chocolate, brownies, cake, even milk has sugar in it. But what about the protein? What about the fat that's in our diet? You have an avocado. You may have heard that's good fat. What do you do with all that? Well, today I'm going to show you how those macromolecules are metabolized and ultimately help to make ATP as well. So let's just go over what you should know by now. You start off with glucose, C6H12O6. And I want to get some participation from you guys, and if you don't volunteer, you're going to be voluntold. What stage of cell respiration does glucose enter? Any volunteers? Yes, Suspina. Huh? What's it? I can't hear you. Yeah, at the very beginning of cell respiration, where does glucose enter? What process? No, not the prep reaction. Before that. Elizabeth? Glycolysis. Remember, this is anaerobic. It's outside the mitochondria. Okay, now, what energy is made by glycolysis? Well, how many ATP molecules are made during glycolysis? Lydia? 2. 2 ATP molecules. How many NADH molecules are made during glycolysis? Alexa? 2. Okay, and then what is glucose broken down into? What is the name of the sugar, and how many of them is made through glycolysis? Mary? Mary? How many? 2. Okay. What is the next metabolic process that pyruvate will enter after glycolysis? And someone last period made a very good point. This is considering oxygen is present, so you are moving into the mitochondria. What is the next metabolic stage of cell respiration? Anyone? Natalie? The prep reaction. How many times do you have to go through the prep reaction if you have 2 pyruvate? Yep, you have to go through it twice. Okay, and let's talk about the products of the prep reaction. There is no ATP made, but there is NADH made. How many NADH molecules are made during the prep reaction? Let's see. Anna, can you tell me? How many NADHs are made during the prep reaction? When you have 2 prep reactions? 2 is correct. Okay. What are the end products of the prep reaction? 2 pyruvate go in, 2 what come out? Lydia? Yeah. Well, that's a waste product. You're not wrong, but what is the intended product? CO2 is a waste. How many? 2 of them. Acetyl coenzyme A. You will then move on to the next metabolic stage of cell respiration. What follows the prep reaction? The Krebs cycle. How many times will you have to go through the Krebs cycle if you have 2 molecules of acetyl coenzyme A? Twice. And you actually make quite a bit of stuff here in the Krebs cycle, how many ATP molecules are produced when going through the Krebs cycle twice? Trey? No, not 4. 2. Do you say 2? 2 ATP. How many NADH molecules are made when doing 2 rounds of Krebs cycle, Lydia? 6. Somebody kept up with her work. You're going to hear that on the video if you watch it. Now, how many FADH2 molecules are made when going through 2 rounds of the Krebs cycle? Annabelle, can you tell me that? 2. Okay, guys, and then what stage am I missing for cell respiration? We got glycolysis, prep reaction, and Krebs cycle. What am I missing? Yeah, well, we're going to do the electron transport chain right now. But what I want to do is I want to get a head count of everything that we have created or synthesized. So let's do NADH first. We have 2, 4, 10 molecules of NADH. For FADH2, we have 2. And for ATP, we have 4. Where will the NADH and the FADH2 go to? Not yet. They're going to the electron transport chain first. They need to have all their energy drained. And FADH2 is going to the electron transport chain as well. Okay. In the electron transport chain, by now, you have a total of 10 NADH and 2 FADH2. This is going to result in, ready? 34 ATP molecules. This form of ATP synthesis through the electron transport chain and using that pump, now you can impress me. What is the name of the pump that makes the ATP? ATP synthase. It is an enzyme that is named after what it does. It synthesizes ATP. This is called oxidative phosphorylation. To be phosphorylated means to have a phosphate added. So you have 34 ADP, and they were given a third phosphate to become ATP. All right. So let's do a quick overview, then we'll move into the other 2 macromolecules. Let you guys get caught up. You made 2 ATP from glycolysis. You make 2 ATP from the Krebs cycle. By what process are these ATP molecules made? Good. This is called substrate level phosphorylation. This just means that these ATP were created from the direct metabolism of a high energy molecule like glucose. And then you have the 34 ATP from the electron transport chain. This is oxidative phosphorylation. And that's everything that we have done since last Tuesday, guys. Summed up in about 10 minutes. How many total ATP is this? 38. So when you are metabolizing one molecule of C6H12O6, you get 38 ATP. Not bad, not bad. Let's see how much you get when you metabolize other macromolecules. So let's you all get caught up. All right. Ready? So, tonight, Mark's going to go to Burns, have himself a steak, and they bring out his steak, and it's got a big glob of butter on it, just melting all over the steak. Tasty. What type of macromolecule is he primarily going to get in his body from eating the steak? Protein. What macromolecule is he going to get into his body from eating that butter? Lipids. Let's find out what the energy content is in proteins and lipids, shall we? So this is going to be other metabolisms. I have a question to start us off. Which macromolecule has the most energy per molecule? I'll give you 4 choices. A, carbs. B, lipids. C, protein. D, nucleic acids. Anyone want to take a guess? No one has gotten it right on the first shot so far today. All right, Mark.
Carbs.
No. Lydia?
Protein.
No. Well, now you're down to 50-50. Lipids. Fat. You ever hear Cartman say, "I'm not fat, I'm just full of potential"? No, maybe. There's a lot of energy to be found in fat. When a grizzly bear hibernates during the winter, he's lost nearly half of his weight because he just hibernated the whole time. And rather than go out hunting for food, its body just metabolizes all the fat that's stored in his body. There's a lot, and that was enough. There's enough energy in that fat to keep a bear alive for months. So there's a lot of energy that's stored in fat. So we're going to do fats first. And more specifically, we're going to do a triglyceride. Triglycerides are what nutritionists and physicians would refer to as bad fat. If you went to the doctor and they did your blood work and they said your triglyceride levels are high, that's not good news. This is, LDL cholesterol. It's bad. It's fat deposits in your body. You've ever heard of love handles? That's triglycerides. Those are fat. Well, a triglyceride has 2 different monomers, as I hope you recall. One part of a triglyceride is the glycerol. And the other part of a triglyceride is the fatty acid chains. There's 10 carbons in each of those fatty acid chains, guys. let you get all caught up. And I need to ask you, not ask you, I'm going to tell you a very important fact that every science student, whether it be high school, college, or beyond, and every professional is aware of. And I want all of you to be aware of it too. So I'm just going to let you get caught up, and I'll tell you this little secret. All right, here's the secret. Well, here's not the secret. You all should know this. How many bonds can one carbon atom have? 4. How many bonds does it look like each of those carbons have? 2, right? Well, gee, where's the other 2? Well, this is a fatty acid. And so fatty acids are carbons with hydrogens bonded to them. But listen up. You don't need to write this. Just pay attention. Just pay attention. It is a given that those hydrogens are there, but why do you think I'm not going to write hydrogens on all those carbons? Any idea? It's actually a lot simpler than you think. You know it's there, and also it's time consuming. I am not going to waste my time going hydrogen, 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 hydrogen. Just know that they are there. And if you go to the very end, the terminal carbons, how many hydrogens can they hold? How many bonds is that carbon at the end right now? That's 1 bond. How many more should it have? So how many hydrogens will be around it? Right. Okay, here's another question from Unit 1. Are those saturated or unsaturated fats? How do you know they're saturated? By looking at it. They're all single bonds. Okay, so guys, here's the first thing that you will need to do. You have digestive enzymes that are capable of breaking the glycerol from the fatty acids. That's the first thing that has to be done. So we're going to metabolize the glycerol first. And I'm going to show you what that will end up as. And then we're going to metabolize the fatty acids, and I'm going to show you what that will end up as, okay? Let me just check something real quick. All right, let's do this. So first off, glycerol is going to be metabolized into 1-pyruvate. What part of cell respiration does pyruvate enter? What stage? How many prep reactions will you have to do if you just have 1 pyruvate? How many NADHs are made through one spin of the prep reaction? 1. Okay. What is the end product of the prep reaction? What molecule is pyruvate broken down to? How many? You have 1 pyruvate, so 1 acetyl coenzyme A. I just spelled that wrong. Okay. What is the next metabolic stage of cell respiration that acetyl coenzyme A enters? How many times will you have to do the Krebs cycle? Once. Okay, when you do 1 spin of the Krebs cycle, how many ATP are produced? 1 ATP. How many NADH? 3. Good. How many FADH2? 1. Okay. We will come back and we will collect those electron carriers after we do the fatty acids, but the breakdown of glycerol is complete. Glycerol is broken down to pyruvate. Pyruvate enters the prep reaction. Then that is broken down to acetyl coenzyme A, and that is going to enter the Krebs cycle. Now let's break down the fatty acids. But before I break down the fatty acids, here's what I need you guys to remember. Check this out. This is just going to be a little reference point. You ready? a fatty acid, can go to, or can be broken down to acetyl coenzyme A. This process is known as beta oxidation. Now, jog my memory. How many carbon atoms does how many carbon atoms are in an acetyl coenzyme A molecule? 2. Okay, now that we have that known, check this out. Look at all that carbon. Oh boy. You can make a whole bunch of acetyls from there. You ready? 1, 2, 3, 4, 5. Tell me again, how many carbons are in an acetyl? How many carbons at a time am I circling? All of these are going to be turned into acetyl coenzyme A's. 2, 2, 2, 2, 2, 2, 2, 2, 2, 15 total. From those fatty acids that I drew, there were 30 carbons. That means that there's 15 pairs. That means you can get 15 acetyl coenzyme A's. This is actually a small triglyceride. Normally, the fatty acids are about 30 carbons long. I only made this one 10. So let's see what happens. We have 15 acetyl coenzyme A's. 15. What step of, make sure I had this right. Check my notes real quick. Yeah. Oh, I forgot to mention this. This is my fault. Come back over here. My bad. When you convert a fatty acid into an acetyl coenzyme A, it makes 1 NADH and it makes 1 FADH2. I forgot to mention that. That's going to be pretty important. So when we have these 15 acetyl coenzyme A's, this is going to result in 15 NADHs, and this is going to result in 15 FADH2s. That's a lot. That's already more than you make from glucose. What step of cell respiration does acetyl coenzyme A enter? Krebs cycle is correct. How many times will you have to go through the Krebs cycle? 15 times. Now, think about this. If one turn through the Krebs cycle makes 1 ATP, 3 NADH, and 1 FADH2, how many ATP are you going to make through 15 cycles of the Krebs cycle? 15 ATP. How many NADH will you make through 15 turns of the Krebs cycle? 45. And how many molecules of FADH2 will you make? 15. All right. So now that we have all of that, let's collect it all. Let's do a final head count and send it over to the electron transport chain, okay? So let's count up all of our NADH, all of it. 1, 2, 3, 4, 15, and 45. So what is 15 plus 45? 60. 60 plus 4 is 64. So all of these we're going to have 64 ATPs. This is all in the electron transport chain, by the way. How many FADH2s do we have? Well, we have 1 here, 15 here, and 15 here. So 15 plus 15 plus 1, 31. Did I put 64 ATP? Darn it. 64 NADH. Sorry, guys. 64 NADH. I don't know why I wrote that. Then we have the FADH2s. That's going to be 31 FADH2s. And now for the final reveal. You have 64 NADHs. You have 31 FADH2s. This is going to result in 236 ATP. Wow, that's a lot from fat. This is all done by oxidative phosphorylation. If you go back up and you count how many ATP you made from the Krebs cycle for glycerol and the Krebs cycle for the fatty acids, that's going to be 16 more ATP. So you add this 16 ATP from substrate level phosphorylation. This is going to result in, I believe it's 252 ATP. That is a lot of energy that you can get from breaking down fat. And that was actually a small triglyceride molecule. If you had a true size triglyceride, it's probably going to be around 600 to 700 ATP. Okay, so I think it's safe to say that we get a lot more energy from fat than we do sugar. But here's someone last year said, well, how come my football coaches don't feed us a bunch of fat instead of like pasta before a game? All right, well, to answer that, carbohydrates are short-term energy, short-term, like 72 hours tops. Lipids is long-term. Remember that a grizzly bear lives off the entire winter off of its stored fat deposits in its body. Also, your body has to be conditioned to burn fat. Let's say that Trey is a triathlete. He goes on Bayshore every single day and he runs 4 miles every day. It would be safe to say that his body is conditioned to burn energy. And so I could give him a bucket of lard and his body is going to break it down pretty quickly because his body needs the energy because he's a well-conditioned machine. Well, let's pretend that Mitchell is a couch potato. He goes home and watches Dr. Phil and Judge Judy every day. He's going to get a bucket of lard, and this just is going to, ruminate and store inside of him because his body is not conditioned to need to burn energy. He's a couch potato. This is why your coaches at the beginning of the year have you running your brains out because they want to get your body into a state where it is conditioned to need to burn energy. And that means that you're going to be used to be able to eating almost anything and it will still be converted to energy. There was a football player a long time ago, not long time ago, but he played for the Cincinnati Bengals. His name was Chad Johnson. He was a big loudmouth wide receiver. And they had a camera crew follow him all during training camp one day. And this guy was ripped, like not an ounce of fat on him. They showed him at McDonald's ordering the whole menu. And he was trying to tell everybody watching at home, next time they tell you can't eat McDonald's, you remember this. And they showed his offensive lineman teammates just eating a banana because one of the conditions in the NFL is if you're overweight, you can get cut. So sometimes linemen come in overweight and they need to lose weight to make the team. And so Chad Johnson is mocking his offensive lineman teammates. Look at these guys. He's eating a banana. I'm over here eating the whole McDonald's menu. He was a professional athlete. He gets paid to run and catch and work out. So he can afford to do that. But his body is used to taking whatever crap you put into it and metabolizing it into energy. Whereas if he's someone who just lies around and watches television all the time, their bodies are not used to that. And so that fat is not going to be metabolized into energy. It's just going to be metabolized, or it's not going to be metabolized at all, actually. It's going to be put into storage, which for us would be like fat rolls, love handles, things like that. Okay, let's do the last one for today. Proteins. What are the monomers of proteins? How many different amino acids are there in nature? There are 20. Some amino acids are big, some are small, some are moderate sized. Well, the first thing you have to do is proteins must be metabolized to their amino acids. If you do not remember what an amino acid looks like, I'm going to jog your memory. Let's see what you guys remember from Unit 1. What is this portion of an amino acid called? This is called the amine group. Remember that Unit 1 material is just going to get harder and harder to remember, so I like to take these opportunities. What is this part of an amino acid called? It has two different names. You're both right. Carboxylic acid or carboxyl. And what is this part? Yeah, the R group gets a lot of different things plugged into it. Well, the reason I drew this is the first thing you have to do is you have to get rid of the amine group. It's got to go. Now, for those of you that either, speaking of working out and getting fit, when you guys go to college, most colleges have a weight room. And a lot of kids end up finding the gym. Actually, a few weeks ago, I found a former student of mine. You guys know Davis Price? You ever heard of him? He went to Plant. I think he graduated last year. The kid was a stick. Some at the gym, he's not a stick anymore. I told him, wow, I'm glad he found the fridge, like in a good way. He's put on a lot of muscle. That sometimes happens. You go to college. You just find the gym and you are growing and you're getting bigger. Well, one of the downsides, I shouldn't say downsides, but one of the effects of having a diet that's very high in protein is that your body produces urea. Urea is a nitrogenous waste product. And it's actually in your urine. It's one of the reasons why urine is called urine, because it has urea. And here's why I mention this. If you're going to have a diet high in protein, like you have protein shakes, you have protein bars, you eat 2 or 3 chicken breasts a day, your body produces a lot of urea. And if you're not drinking a lot of water, this urea is going to accumulate in your kidneys. And guess what that causes? Kidney stones. Yep. So you can avoid this entirely by just drinking a lot of water. If you watch those videos on Instagram of The Rock, Dwayne Johnson, working out in his gym, he has a bucket of water because he's eating a lot of protein. He's a big dude. And in order to avoid getting kidney stones, which can be extremely painful, you need to just keep everything flushed. You need to flush your system out. And so water, water, water if you have a high protein diet. All right, so you take out the amine group, and then what you have left is the R group and the carboxylic acid group. This is where you can get various molecules. So the rest of it can be converted to pyruvate or acetyl coenzyme A, or succinate. Let's see how well you all were listening on Monday. How many carbons does succinate have, and where did we find it? Anybody recall? Really? It has 4 carbons, and it is in the Krebs cycle. Good job. Now, why such variation? Here's why. I'll put it over here. The molecule produced depends on the amino acids R group. Guys, you will find out when we get to Unit 5, which is molecular genetics, that amino acids come in all different shapes and sizes. And it's because of the R group. Some R groups are huge. Some R groups are small. Some R groups are medium-sized. So I want to ask you all, we're coming to a close here. If a amino acid was converted to pyruvate, where would it enter cell respiration? Where would it enter cell respiration? The prep reaction. Good. If an amino acid was converted to acetyl coenzyme A, where would it enter cell respiration? Good. And if succinate was formed from an amino acid, where would succinate enter? What part of the Krebs cycle? I'll give you a choice, beginning or middle. Yeah, because remember we didn't get to succinate until we got into the middle of it. So I'm going to put Krebs cycle, but I'll write middle. Okay, and then you just go from there. You make all the NADH and ATP and FADH2 that you normally would make. So let's do a final overview, and that will be it for today. You have glycolysis, prep, Krebs, electron transport chain. The 4 main steps of cell respiration. Which one is anaerobic? Yeah. All right, so eeny, meeny, miny, Ben. Ben, if we are eating carbohydrates, what stage of cell respiration would carbohydrates enter? Things like sugar, and more sugar. No, not the prep reaction. Glycolysis. Glycolysis means sugar splitting. So if you eat sugar, guys, it's going to enter through glycolysis. Good? Now, let's do fat. Now I'm going to break this up. You ready? Check this out. Glycerol, fatty acids. I'm going to ask 2 different people here. Mariah, if you had glycerol, where would glycerol enter cell respiration? Glycolysis. Not glycolysis. This is what we just did today. Look in your notes. Glycerol gets converted to pyruvate. So where would that enter cell respiration? If it's converted into pyruvate, where would pyruvate get started? Prep. Okay, Mark. Fatty acids. Fatty acids get broke down to acetyl coenzyme A. Where are they going to enter the cell respiration? Correct. Okay, and then lastly, we have protein. We break this up into 3 parts. Pyruvate, acetyl coenzyme A, succinate. Amaya, where would pyruvate that is made from protein enter cell respiration? Correct. Mitchell, where would acetyl coenzyme A that is made from amino acids enter cell respiration? Correct. The beginning of it. And let's go to Natalie. Where would succinate, which is made from amino acids, enter the cell respiration? The middle of it. Good. Good call. Okay. So as you guys can see, you don't just make ATP from carbs. You make ATP from proteins and lipids as well. Lipids makes the most. And that's it.